Single-cell RNA sequencing is a powerful technique for describing cell states. Unfortunately, identifying the spatial arrangement of these states in tissues remains challenging. We've developed a rapid and integrated method called SEEP, or segmentation by exogenous perfusion, to link positional location to transcriptional identity within 3D disease models. Our method utilizes the steady-state diffusion kinetics of a fluorescent dye to establish a gradient along the radial axis of 3D tissues. As shown in this illustration, we exposed 3D tumor models of high-grade serous ovarian cancer spheroids to a calcine bath. Due to a reaction-diffusion balance of the calcine uptake into the tissues analyzed, cells on the surface of the tumors are exposed to higher levels of calcine than cells deep within the tumors. This stable gradient allows us to correlate cell brightness with cell position and spatially segment tissues into concentric layers. After shell volumes have been defined using a machine learning algorithm to segment confocal imaging data, tissues are disassociated for single-cell characterization. It's at this point that most single-cell RNA sequencing assays lose spatial information, as cells are by definition stripped from their native environments. SEEP is able to keep track of cell positions by sorting and binning cells according to their brightness, and indirectly, according to their distance from the tissue surface. As demonstrated in the FACS profile below, bins are defined according to the image segmentation of the previous imaging steps. After binning according to spatial positions, cells are processed through a droplet-based microfluidic system to allow for transcriptome barcoding, library amplification and indexing, and high-throughput sequencing. The single-cell RNA sequencing data is then analyzed and incorporated with positional data to characterize cells by both their transcriptomic and their regional identity. Tissue regions can then be described by their composition of various transcriptional states. We use SEEP to analyze spheroid, organoid, and in vivo tumor models of high-grade serous ovarian cancer. Our results validated long-standing beliefs regarding the relationship between cell state and position, while revealing new concepts on how spatially unique microenvironments influence cell identity. For example, signatures of hypoxia were found deep within tissue cores, while signatures of cell proliferation were found to be most heightened within the intermediate tissue layers. Less intuitively, we found that surface-residing cells were enriched for transcriptional traits associated with inflammation, immune response, and an epithelial to mesenchymal transition. These signatures have been shown to be beneficial for tumor seeding, invasion, and metastasis in ovarian cancer. But to our knowledge, these have not been reported to be associated with tumor surfaces. We hope that this method will continue to be used and adapted to unveil new insights into tumor biology.